Thank you, Nit. Appreciate everyone turning up. Good afternoon, good morning, wherever you're at. And so today we're going to talk about the Varroa mite. Hopefully, as current or hoping to be beekeepers, it's something that's on your radar because it's really important. So myself and a couple other field specialists are going to kind of talk through this thing that we have to deal with, unfortunately. Here we see this just opening slide, a really uh, close-up picture of the kind of know the enemy. This is what we're dealing with. This is what it looks like under an electron microscope. Just to give you an overview of Be Informed, as Net mentioned, we have been doing a series of webinars. There will be also some coming up in the future. Those are available at beinformed.org/education. You can find all those recordings. Our program, we're a nonprofit based out of the University of Maryland. That's our diagnostic lab, but we work cooperatively with quite a few universities. You can see all these collaborators here. We do kind of have a national reach. We're working with beekeepers across the country. Overall, we're somewhere around about a dozen staff members between our IT diagnostic lab, field specialists, which is what I and the others that are going to talk today with you. That's what we do. Main mission of our program is to improve colony health and reduce losses. We do this by supporting beekeepers, acting as a kind of a conduit or a bridge between science and industry, connecting those two aspects so we can start to solve some of the issues with that we're facing with the bees. In that process, we've kind of become the largest repository of colony health data in the country. We do the annual management loss survey every year. We'll show some results from that today to kind of provide data to support the messaging we're bringing today. So we'll move on from that, but that's to say, I'd encourage you to visit the website. A lot of good information there. So myself, as Net mentioned, Dan Wines, I'm based out of Michigan State University. I'm going to talk through the Varroa mite, a little bit of the history, biology, and life cycle. Ben Sallmann from Oregon State University is going to talk through kind of the population dynamics and growth in the colony, as well as monitoring methods. And then Nelson Williams is going to finish with, he's based at University of Minnesota. He's going to finish with kind of some intervening and control options for the Varroa mite. So, we hear a lot as people that pay attention to bees, work with bees, a lot of messaging out there in the public. Bees are struggling. That's been, sort of well documented for the last decade plus. There's a lot of misconceptions about why, what these causes are. We that kind of work in this field, kind of distilled it down to the 4 Ps. A couple of those kind of occur on landscape level, those things like pesticides, things, ag use and homeowner use. And then poor nutrition, bees derive all of their nutritional needs from flowers via pollen and nectar. So, not having enough and a diversity of floral resources can be problematic for the bees. And then there's kind of 2 things that are sort of more in colony level. Pathogens, bacteria, just like any organism, they have, bacterial diseases, viruses, things like that. And also parasites. That's what we're going to talk about today, and particularly one that is primary parasite, the Varroa mite, which we see this photo left of the highlighted parasite cell. That's a pupa with half a dozen of the mites on it. They are a major problem. They are certainly a huge contributor to colony stress and colony losses, but fortunately, as beekeepers, this is one of the stressors that we are in a position to directly impact and make a difference and help our bees out. This next slide demonstrates that. We can see here. So this comes from, as I say, this kind of management and loss survey. And what we've got here, these vertical bars on the left, this is broken down by scale of beekeepers. So we have backyard, it's kind of less than 50, sideline 50 to 500, commercial greater than 500. And so the yellow portion of the bar, the true, is response to the question, do you treat for Varroa mites? The brown would be a negative response to that. So we can see, if we look at the commercial, almost exclude, nearly all those beekeepers, almost 100% do treat, about 80% of the sideliners do it. But once we get to the backyard, kind of hobbyist, small scale beekeepers, it's about 50% that do treat. Um, on the right, these horizontal bars, same color coding. The true is a yes, I do treat for Varroa. The brown is no, I do not. And it's broken down by these three different scales of beekeepers. The small scale at the top there and the larger at the bottom. We see in all cases, regardless of scale, that beekeepers that do treat for Varroa, that yellow bar, they have significantly less colony losses than those that do not. So again, it's somewhere we can make a difference. There's a lot of bee problems that are complicated and kind of beyond our control as individuals, but Varroa management in your own colonies, we have data to support that it does result in less losses. So we'll move on to just kind of outlining the biology and life cycle. I'll start with just kind of giving you a little bit of the background on this organism, the history, because it does kind of inform the state of where we're at with it and why it's so problematic. So Varroa destructor, it's external parasite. We've seen a couple photos there. We'll see more, but it lives on the honeybees. It was first discovered, documented in Indonesia on the Asian honeybee, Apis ceranae, in the early 1900s. First observed on what we as beekeepers keep in this country, the European honeybee, Apis mellifera. Colonies, European bees were brought to Asia, and in the 60s, this Varroa mite was first seen to have jumped species from its original host of the Apis ceranae to Apis mellifera. Now, this Varroa mite was then seen on bees in Europe throughout the 70s. It had spread across all of Europe. It first arrived in the United States in 1987, first seen in Florida, and then in Wisconsin on packages that had been shipped up for Florida. And that arrival in 87, by the early 90s, it was only a handful of years where it was documented in every state in the United States and contiguous 48 and Canada. The way bees are moved around this country, whether it's for pollination or shipping of packages, things like that. Once they were here, they were everywhere very quickly. And in a global context, that's very true as well. The only kind of major exception where the Varroa mite is not living on European honeybees is in Australia. They have very strong biosecurity. They're very proactive. And to date, they have managed to keep it out. But Australia and a few isolated islands are the only places where beekeepers are not dealing with Varroa mite. This is important when we think about, the honeybees haven't had a long time to kind of, evolve with this parasite and reach a balance. They just don't have any defense against it. So that's where we as beekeepers need to intervene and need to help them along. Now, with the Varroa mites, they have kind of two different stages. They're either living on the adult bees. We can see this one, picking it up here with picking up the bee, and you see a mite on the abdomen there. It kind of gives you a sense of the scale of that parasite relative to its host. That's quite large in the realm of parasite-host relationships. So they're either living on adult bees or they're going to be present in the brood, as we see in this middle photo, and that's where they reproduce. You can see this on the right. We'll get a close-up of this picture next, but it's essentially a tub full of mites that we get when we do monitoring methods and alcohol wash, which Ben's going to talk about. Here in this tub, we have, I think it's 160 some mites in this tub. We've done an alcohol wash on about 300 bees. We're at a huge infestation rate here. Just to little bit of reference, if I get to double digits when I'm counting, somewhere 10, 12 mites, for 300 bees, we're up in 3-4%. I have serious concerns about the future health prospects of that colony. So certainly when we get into triple digits, we've got big red flags here that colony is struggling and will likely continue to struggle. So we see here in this tub, these are all the mature females. On this next slide, we've kind of got the cast of characters, the little who's who in the different stages. But when we count mites, we're counting these mature females. This top left photo, they are bigger, they are crab shaped. They kind of have this dark red burgundy color. The male mite below we see is much smaller, kind of a tick shaped. And then to the right, we kind of see the developmental stages of the Varroa mite. The next slide we have here shows them kind of all, we call this family portrait. It's a photo Ben took. And it's, the cell's kind of been opened up. Looking down into the cell, we see the red mature females. We see immature female, we see a male, we see an egg. We also, one thing to note here on the top of the cell, we see this kind of white crystals. That's with the cursor there being highlighted. That's mite frass, and as beekeepers, that's something we should be looking for as we examine our colonies. It's not a standalone monitoring method, but it is something good to know what it is and keep your eye out for. So, to walk through the life cycle, basically, we have these mature females, and the foundress mite enters the cell. So we've got a series of photos here that's kind of going to show you what happens. And it'll give you the progress essentially of mite reproduction, and we'll talk through that over these next handful of slides. So what we have, as we said, we have kind of these mites existing in two different states. Mites on the bees, and what's going to happen is a mite's going to jump off of an adult bee and into the cell. This typically happens right before capping. They're able to detect the brood of the appropriate age, which right before capping. They get in, they crawl right under that mite, so they're very well hidden. They actually have a little kind of snorkel-like appendage so they can breathe, but they're kind of hiding under there, avoiding detection until capping occurs. The next thing on this next slide, we see, so this larva is pupating, and that foundress mite, that mature female that's entered the cell, actually kind of chews a hole into that pupa. She's got some kind of incisor appendages that she's able to penetrate that bee, and she's feeding off it. And then she will begin laying eggs. Again, this is happening all under the capping. After about 60 hours, so middle of 3 days after capping, somewhere on that third day, she's going to lay, the first egg she lays is going to be a male. And then approximately every 30 hours after that, she's going to lay a female egg. So there will be kind of a continuous stream of female eggs. This is all, again, happening underneath the capping, and those are all feeding on this developing pupa. Next slide here, we kind of see, we've got our mites developing there. We've got males, we've got a couple females, and they're as they're feeding on this pupa, they're all kind of perforating this developing pupa, and it's really problematic, and they're doing mechanical damage. They're essentially chewing holes in this developing bee, but they're also spreading viruses. And this is one, we as those that work with bees, bee scientists, beekeepers, we don't have a thorough understanding of the viruses, but we know they are very problematic. It's kind of an emerging field in the recent years of how bad they are, and this is really kind of amplifying the impacts of the mites. And so these viruses, we don't have direct ways to control them. We do know many of them are spread by mites. So our best course of action is kind of preventing the viruses from getting a hold by keeping the mites at bay so they cannot vector those viruses and really do long-term damage to colony health. So as this is all happening, mites developing, next slide, we have, females emerging, and they essentially, as this bee is developed, it's going to chew out from under its capping, and along with it are going to come with it mites. If it's a worker bee, we'd expect one or two of those daughter mites in addition to that foundress, that kind of mother, coming out from the cell. Any mites that have not had long enough, that third, fourth, fifth female egg laid, is not going to develop to maturity and will just die, unless it's a drone cell where there's a longer development period and we can have more mites coming out. So when those mites have emerged from the cell, on this next slide, it kind of starts the process over. That mite is going to come out with that bee, and it's going to jump on another bee, and eventually it will go back into a new brood cell just before capping and start that process over. Now, Net mentioned we did have some questions come in prior to this webinar, and one of them is kind of relevant right here. I'll address it. The question is, how long can Varroa live for off of the adult bee? I asked because there's been no brood present in this colony for a few weeks, and I did see the mite in the bottom of an empty cell whilst doing an inspection. It made me wonder how long of a brood break was needed to reduce number to a level the bees could tolerate. So kind of multi-part question. Short answer, as far as how long can mites live on adult bees, several months. I don't know the upper bound of that precisely, but Mites do overwinter in the winter cluster of the bees when they're in brood this period. You know, where I'm at in Michigan, that's probably about 4 months or so, from late October through some point in February, depending on the severity of the winter. And some mites will come through with those bees, so they are able to live for months and months. Now, the idea of a brood break, you do hear that as a potential mite management method. Um, what's happening there while we just showed they need to reproduce in the brood. So with no brood, we are stopping their reproduction, but we're not halting or we're not decreasing the population in any meaningful way. There will be some mites that do die, but that's not a brood break alone is not going to be sufficient for a control method. When we talk about a brood break, it's a point in the peak active brood rearing season. If there's a brood break through a queen event, it's really creates a kind of a point of leverage where mites are easier to kill when they're not under the cappings. And Nelson will kind of talk about a little of that when he talks at some of the treatment aspects. But that's what we talk about as a brood break in regards to mite management. Just having them go queenless is not going to be sufficient to reduce mite numbers, and your colony is just going to dwindle and die if it's queenless for months on end. So that's not a viable standalone option. So just to look at kind of the math on why this is problematic, let's say we have, under this capping, that foundress mite, her first daughter is going to be mature and mated after about 10.5 days under a capping. Now, if we look at the development of workers, we know they emerge 21 days after being laid. They're capped on day 9, so they have about 12 days under that capping. So we're certainly going to get one new mite plus the foundress, and quite often we can get 2 mites out of that. So about 1.5 per is kind of the average. Where it gets really scary and populations build much quicker is we know drones are 24 days versus 21 days, yet they're capped at the same time. So they have the full 3 extra days under the capping. And that means a couple of those 3rd and 4th female eggs have time to mature into mated mites that can then become foundress mites of their own and carry on. So this mite population growth is really exponential, particularly at certain times of year, which Ben's going to talk about why that monitoring is really critical. We'll just move into a couple things, having just to say basic background on the reproductive cycle. We'll talk about why this is so problematic just with a few photos here. We really do need to help our bees deal with the Varroa. They've only had a couple decades, and they've not evolved behaviors and through genetic selection to kind of reach a balance with this parasite. Varroa mites, they flat out kill colonies. It's not a quick or clean death. It's a long suffering dwindle. I've got plenty of ugly photos of sick colonies that are preventable from mite damage. There's also an aspect of being a kind of a good beekeeping neighbor. As that colony collapses and dies, those mites don't disappear. They're going to end up in neighboring colonies through drift or robbing. Could be in your apiary, could be miles away. So to kind of be a good member of the beekeeping community, it's an obligation to manage the mites. So in addition to the animal welfare aspect. So had another question come in here regarding kind of bald brood. A question says, is bald brood often a sign of Varroa beginning to get a hold? Is this a way that some bees disrupt the Varroa feeding in the pupa? Short answer is yes. When we see uncapped cells, like we see this photo here on the left, there's a lot of uncapped cells. And that is an indication. It's not necessarily Varroa mites, but it's something called hygienic behavior. It's when the bees chew open a cell like that, they've detected something wrong. Hygienic behavior will also, bees will detect things like chalk brood or American foul brood, but they're detecting problems. The bees chew open that cell and begin removing it. Now, this photo on the left here, we see kind of several stages, and it's important to note we see development, we see recognizable body parts, we see eyes and antennae and heads and abdomens, and so some have been chewed out more than others, but it has pupated. On the right, we see this in the middle, this cell that's been uncapped, it's kind of in the pink eye or purple stage, and we see a mite right on top of it. We know it has at one point been capped and then reopened because it has pupated. And just another thing to kind of draw attention to, we look at the cells on the right photo, the cells above with the cursor highlighting there, that's mite frass. That's when we see, there's, we're looking in a handful of cells here and most of them have mite frass on the top. So this colony is really carrying a high infestation load. But again, on this photo, notice the defined body parts. We see pupation has happened. Now, when we go to the next photo, on this next slide, this is something a little different happening here, still mite related, but different kind of visual presentation. We call this parasitic mite syndrome. And we see here, we kind of have this just kind of white goo. There's some little bit of yellow tissue that's mid-gut. And these have not pupated. They've kind of just, broken down, melted. They've died before that stage of pupation. There's no discernible body parts, but it is mite related damage in this photo. And then just lastly, there's another photo here of actual damage to the adult bees. We've kind of seen what it does in the brood. Here, this left, actually both these photos, these bees have deformed wing. This is, I mentioned, the mite spread viruses. You end up with this particular virus deformed wing. These little bees have twisted up wings. Clearly, it's not going to fly. It's not going to have a long life. It's not going to contribute to the colony's health. It's actually going to spread viruses itself through just being in the colony in contact. And, on the left here, we even see a mite hiding right between the abdomen and thorax, just in the triangle of those wings there. So, say mites do damage to brood, they do damage to adult bees. When we think about colony health, we're thinking about the whole colony. So just as we're going to kind of transition, we can see these things do damage colonies. We're just because we're dealing with kind of a colony of insects. It doesn't mean that we don't need to have their health in mind. We think nothing of treating cats and dogs for fleas and heartworms and things like that. Certainly farmers, treat cattle for ticks and things like that. The honeybees deserve the same level of care. I think I've kind of tried to lay out a little bit of the biology here, and now we're going to kind of move into with Nelson and Ben, talking about knowing when to intervene through monitoring strategies, and then knowing how to intervene, talking about treatments and what we want to do when we determine we need to take action. So we'll pass it off from there, and we'll answer any further questions on this topic at the end.
All right, thank you, Dan. My name is Ben Sallmann. I'll be taking care of this next section here on growth and monitoring of Varroa. I'm the field specialist for the Pacific Northwest region, based in Corvallis, Oregon. Previously worked in Northern California with the queen breeders, so I've been up here for 3 years now, working with these beekeepers in this region. And believe me, we see plenty of mites in all the regions. So to start off here, we're going to ask a quick poll question. It should be popping up soon here.
So if you all could hear me, for the sake of the recording, I'm just going to read what the polls are. This initial one, we would like to gauge our audience for these sessions. We have over 200 participants right now, so we want to just get an idea of who's logging in. Give it another 10 seconds and then we'll move on to the Varroa related poll question. All right, we have 75% of the people responding. I'm going to share the results real quick. It looks like we have about 150 people out of that set as backyard at 88%, sideliners at 8%, a commercial beekeeper has logged in, and there's about 4% of folks who are just listening in. They're not currently keeping bees. Let me go on to the Varroa related poll. Okay, so these questions are asking about how often you monitor for mites. I'll give it another 15 seconds. right, I'm going to end the polling. We have a little over 80% responding at 10% once per year, twice per year at 32%, at least 4 times per year at 27%, every month at 20%, and I do not monitor Varroa at 11%.
Great. That's actually quite encouraging to see so many people are actually monitoring at some point during the year. According to our survey, as Dan said, there's almost half the backyard beekeepers don't monitor. So it looks like we've got a good audience here that's on top of their Varroa monitoring. Going on, I'll just talk a little bit about the population dynamics going on in the colonies relating to mites and honeybees together. So as you can see in this chart, the bee and mite populations both start low in the beginning of the year. This would be in your, very early in the spring or over winter, where the bees and mites are dormant. And then as the queen starts to ramp up and starts to lay, the population of bees really increases much faster than the mite populations. So they can outrun any mites that are there if the queen is doing her job and the colony is healthy. You can actually go the first half of the year without seeing too many mites. They're definitely there. They're mostly in the brood. And what really causes problems is later in the summer or later in the fall, when the bee population starts to level off. The mite population is still increasing. So the ratio of mites to bees really ramps up later in the summer, and there's less and less brood for the mites to crawl into. So any brood that is remaining, in the fall, if the mites are still real high, you get multiple foundresses going into the broodettes of the right age, and you can if you have any kind of viruses, that's when they really explode, and can that's when you see these colonies collapsing. Is later in the year when maybe the beekeeper didn't see many mites throughout the summer and they thought they were good to go without treatment, and then they get a nasty surprise in the fall. And that's also how mites are mostly they're incestual. They mate with their brother, but in this case, when in the fall, when you have multiple foundresses going into that limited amount of brood, you can get mites crossbreeding with each other, and the mites are able to have that genetic variation in their populations. So the key here is also it doesn't really show up on the graph here, but right in the middle of the graph there, I think that mite population, if the colony is big enough, that it has enough resources to make drone brood, that's when the mite population can really skyrocket, and the rate of increase can really pop right there. So this doesn't really show that. But I think in a smaller colony, a new split that doesn't have a lot of extra resources, a lot of times by the end of the year, their mite levels will be much lower than the mother colony that never was split, that started off strong, because they had a lot of drone brood. So that's just something to keep in mind. We often see the new starts, new packages started in the spring. Their mite levels tend not to get as high as the colonies that started off as beasts in the early in the year. And then going on to the next slide, there's a couple websites that are pretty useful that have mite models where you can plug in the different variables in your colony, the frames of bees, any kind of queen events, the forage situations, and then treatments, and then it'll model your mite numbers throughout the year. And this is something that I'm not going to get in the weeds and pull this up, but if you have time and are interested, they're actually pretty fun to play with. Scientific Beekeeping also has a mite model on the next slide here, where Randy created this mite model. That's I was playing with it a little last night, and it's pretty easy to use. One thing that really came out to me was messing with the treatments. You can see the beginning of the year, the blue line is the actually mite levels in the alcohol wash. It seems real low, in most of the spring. That's because those mites are in the brood. And if you can throw any kind of roadblock in front of those mites and slow them down early in the year, that even if it's a softer treatment, you don't have to get down to zero to really make a huge impact later in the year, where they start to exponentially skyrocket their And this chart shows a colony that has not been treated and what happens with those mite numbers in the fall. Starting in, you know, September, October, the ratio of mites to bees really starts to get out of control, and that colony is probably a goner. So again, if you have time and interest, I would recommend looking at these and maybe plugging in your ideas for management strategies and see what the model pumps out. It's worth looking into. And next slide. We have another poll here asking about your monitoring.
All right,
So
poll is open. We'll give it another about 25 seconds here. Let's aim for 75% reporting. Let's see if we can get there. All right, perfect. I'm going to end the poll and share the results. It looks like alcohol wash wins at 52%. And then we've got visual inspection of adult bees at 31%, visual inspection of drone brood at 27%, sticky board or mite drop at 38%, powder sugar shake at 35%, eat the roll at 1%. And just note that participants were allowed to select more than one answer. So some folks use more than one.
Yeah, this is great. This is encouraging. This looks like most of you are monitoring for Varroa, and over half are using the alcohol wash, which is our preferred method. I'll go into why that is in a minute here. We don't recommend just relying on a visual inspection of the colony. It can really be easy to miss a mite problem. The mites, if you do see a mite on a bee on its back, you're seeing a tiny fraction of the total mites in that colony. So most of the mites are going to be under the cap brood reproducing, especially, you know, in the first half of the year. And then when those bees do hatch, the mites tend to be clinging on the underside. They're in the sclerites wedged in there and feeding. So it's not that common for the mite to crawl up on the back of the adult bee. So if you're seeing mites on bees, you have a high infestation, and it's important to monitor before you get to that point. So some beekeepers will crack the brood boxes open and look at the drone brood in between, and that can tell you if you have mites present. But there's been times when I crack that open and see no mites in the drone brood, and then some mites show up in the sample. So it's not foolproof. But if you crack that drone brood open and you see mites crawling over, you know, it's immediate sign that they're there and reproducing quickly. We don't recommend the ether roll as much either. That doesn't capture enough of the mites. So you know, 50% or so. And if you're right on that threshold, you know, it can make the difference between feeling like you're okay, safe, good to go, or you need to treat over the threshold. So like a more accurate way to count the mites. The sticky boards or a mite drop, this has some value, but it mostly tells you if you have a lot of mites or a few mites. It doesn't, without context, the information is not as valuable. And there's a couple ways to do this. You can do a shock drop where you put in a really hot treatment and see how many mites drop off onto the the sticky board on the bottom of the colony. Or you can do a natural drop where you just put it in there for 24 hours and see how many mites are just falling. Some commercial beekeepers will use this after a treatment just to see if they can drop more mites after the treatment, which would show that they need to treat again. But our preferred method is the alcohol wash. We've done some trials and found we get well over 95% recovery of the mites. The powder sugar shake is another option, especially for beekeepers that don't like the thought of killing bees. We don't recover as many, but it's definitely better than nothing. And I have a feeling that a lot of those bees that come out of the sugar shake, they don't survive much longer anyways, because if you really want to get the mites out, you have to shake vigorously for a minute, and that does, I think it does, you know, it reduces the lifespan of those bees. So if you really want to get an accurate count, I feel like alcohol wash is the way to go. So here's some data from our loss survey, and it shows the different monitoring techniques in the left there. So for commercial beekeepers, over half use alcohol wash, or they have us come out and do the alcohol wash for them. There's a few that do the mite drop, and visual inspection. It should be part of everyone. It just not can't be relied on 100%. Sideliners, are kind of in between. In the backyard hobbyists, you see only a few are doing alcohol washes according to our survey, where a lot are doing the mite drop and the visual inspection, and a good percent don't monitor at all. And you can see in the right there the levels of loss associated with those different monitoring techniques. The alcohol wash tends to have the lowest loss rate for those beekeepers that use that. Also in that bottom right figure, it does show that commercial beekeepers using the mite drop, they have the lowest loss, but again, that's probably in conjunction with other mite monitoring, and they might have more context to look, when they're looking at those mite drops, they have a system in place that's been working for them. So moving on to the next slide. We're going to show a quick video. This is Dan Arell, our Texas field specialist, or former Texas field specialist. He has done a lot of these alcohol washes like we all have, and we're going to see how he collects the bees for the sample. So we can go ahead and play that video. So I'm not sure if you're getting sound here. I'm not getting any sound, so maybe I'll narrate over this.
The collection method that we use for the alcohol wash way of checking for varroa mites. So here I've got my honeybee death chute. I've got my frame of brood with at least a decent patch of open brood. So this is plenty of open brood for an alcohol wash sample. I've seen the queen in this hive, and I know she's safe on another frame. If I hadn't seen her, I would take another close look once the bees are here on the trough. But now that I know the queen's not on this frame, I'm just going to go straight ahead. That's a level quarter cup. And I might need a few more.
Dan's using 2 quarter cups. You can also use just a half cup and make sure it's tamped down to get the good measurement.
Another level quarter cup. And there are the samples taken.
Bees.
And here's a chance to look at a frame that, once the bees are all off it, you can take a closer look at the health of the brood. This looks quite beautiful. And we'll close her up.
The reason you want to get open brood for your sample is you want to get a number of the young newly hatched bees, where the mites, if you're going to find mites, you're going to be on those younger bees that have just hatched, and the open brood, you'll see the nurse bees are going to be there. Also, there's usually some sealed brood that's nearby that's freshly, it's hatching out as you're collecting the sample. So, I mean, if you want to find mites, and that's the whole point, it is important to get that sample off of a brood frame and not off the honey super. I mean, you find some mites on those older bees, but most of them are going to be on the newly hatched bees. So Dan took 2 quarter cup measurements. That's the way he likes to do it. The goal is to get as close to 300 bees as possible. I think a half cup measuring cup works fine. And as you're taking that sample, you'll have some of the older bees, they'll be flying off, and that's fine. And you just want to make sure that you have that full half a cup or 2 full quarter cups. And there's other ways to do this. You can also shake that frame into a tub, making sure that the queen's not on there, and then let some of the foragers fly off. The young dumb bees will be left behind. They won't know where to go. So will just be sitting in the bottom of the tub, and then you can scoop those up. And that's another way to collect the bee sample. The next video will show their actual alcohol wash procedure. We all have little variations on this, but this is generally how we do it.
All right, I'm Dan Orell with the Bee Informed Partnership. Just gonna give a quick demo of how we do our field alcohol washes, which is quite a reliable way of measuring the mite level in colonies. So here I've got 2 samples in isopropyl alcohol that we've previously collected. It's about 300 bees measured by volume by shaking off a brood frame of bees into this collection chute and taking 2 quarter cup measurements. Isopropyl is about 30 to 35% strength. So the first thing we're going to do is shake these for a minute. But first I'll just show you what I have in front of me. I have these original collection items. I've got my mason jars more than half filled with alcohol with the nice plastic lids. I've got a stopwatch for the, or a wristwatch for the, to time the minute shake. 2 bins. I've got a wide screen that will let the mites fall through, but keep the bees on top. And I've also got a fine screen that'll let us strain out the mites. All right, so here goes a minute.
So Dan here is using 35% alcohol, and we found that it's this year because of COVID, it's been really difficult to get alcohol. The stores are sold out a lot of times. So there are some alternatives you can use. Windshield wiper fluid works well, but make sure you get the winter formulation that has a higher alcohol percentage so it doesn't freeze. You want at least 30% alcohol in those formulations. And if you can get rubbing alcohol from the pharmacy, usually that's sold at 75% strength. So if you just cut that with water in half, that'll be a good percent of alcohol. There's also some work being done on using just soap water and not agitating as much as swirling, and that seems to be somewhat promising. I still think alcohol is probably the best. Those mites get dislodged pretty quickly with alcohol.
All right, that's our minute. So what we're gonna do is we're gonna pour these bees through the coarse sieve 3 times with strained alcohol each time. And in between, so after we've shaken, so between each sieving, we're gonna turn the bees. All right. So I'll do the first bottle fairly slowly. First, I'm gonna swirl the alcohol to suspend any mites. Pour them out on the coarse screen. I'm gonna turn the bees. Filter the alcohol through this fine sieve. Give the bees a second rinse. That was my second rinse. Turn the bees. For the third rinse, now we filter that alcohol back into the jar and we can use it again.
So then there's counting the mites in the sieve. You can also count the mites directly in the tub if you pour the alcohol through. You can they're easy to see up against that white plastic.
Second one a bit faster.
And when he tosses the bees, they should be pretty dry. You notice he'll shake that sieve, and the mites will fly out of there. You want those bees to be dry, not soggy when you're done with them. And if you're not that familiar with identifying varroa mites, once you see a few, you'll really get the hang of it. But they're really distinct color, this reddish brown color, and they're very specific, distinct size. So once your eye is trained, they pop right out
There
at
are
you. You
mites.
don't count the debris as mites. There can be little bits of pollen or bee parts that are in there
Thanks for
floating around.
watching.
Sometimes you'll even get fruit flies or other insects that like the smell of alcohol. They'll dive into your alcohol and they'll show up in your alcohol wash, and you have to make sure to not count those. So, and also remember when you got your mite number to divide by 3, that'll give you your mites per 100 bees, since the sample was 300 bees. So, coming up with 12 mites will be 4% infestation. Okay. Next, I'll just quickly go over the thresholds for Varroa. I feel like when Varroa first came on the scene in the 80s and 90s, you could carry really high mite loads and the bees weren't that affected. But as the viruses have become more virulent and problematic, the threshold, I think, is going down, and the treatment needs to happen earlier than it used to. I remember seeing bottom boards just full of mites after the first, the first Kumafos treatments came out. It was effective, and you'd see just the coating, the mites coating the bottom board. These days, you just can't get away with that kind of infestation. So as the colonies are growing in the spring, there's a lot of brood. You shouldn't be seeing a lot of mites in your samples, because most of those mites are going to be under the brood. If you start to see a threshold, if you start to see mites over 1%, getting 2% in the spring, that's going to be an issue later. That mite population is going to really explode in the summer. So it's important to keep those mite levels low in the spring. They don't have to be 0. If you have 1 or 2 mites in a few of the samples, it's acceptable. Where it's really crucial is in the fall, when all that brood has been is hatched, and most of the mites are going to be on the bees. But you want to have a good fall treatment planned. We have the threshold as 3 mites per 100 bees in the fall. I think most beekeepers would be happier with a lower number, as close to 0 as possible. Because if there's mites in that cluster over winter, they're going to dive into the first round of brood in the spring, and they're going to mess up your first round of foragers and nurse bees that are going to get that colony going. So I've seen colonies in almonds that went into winter with high mites, and they never get off the ground in the spring because that first round of brood was infested with mites. So there's still a little bit of debate about controlling or not controlling Varroa. There's some treatment-free beekeepers still trying to do it out there, but we don't recommend that, of course. You're going to end up with a mite bomb, where the colony is just collapsing and exploding and spreading mites everywhere, either from robbing or just those bees absconding and getting into other colonies. And that's just bad news all around. And then the viruses really can explode. And once those viruses get a foothold in the colony, it takes a while for them to get out. It takes some turnover of bees to end with low mites or no mites to get healthy brood again. So it's best to not let the viruses get out of control from the beginning. So these are some of the arguments for not controlling. Is we hear a lot, well, I didn't see any mites, so I don't have mites, and I don't need to treat. And that's it's an easy mistake to make for a beginning beekeeper, but it's really crucial to monitor their mites. If you haven't been treating, you have mites. It's almost guaranteed. So knowing your mite levels, monitoring is the first step. There's some beekeepers don't want to use chemicals. They would like to do all natural organic beekeeping. And again, I understand the intention. I think it's a good intention to have, but again, if you want to be organic, there are some organic treatments, organic acids that are non-synthetic, and you can also do some of the other drone removal in addition to your treatment. So there's ways to control the mites without synthetic chemicals. The important thing is to do something. Another reason people don't treat is they think that they're helping breed a stronger bee, survival of the fittest. If their bees die, it means they were just weak. But that's just not. I don't see that as being effective. If you think about human health or anything, you might survive, but if you're sick and not thriving, then what's the point And if you have bees and you're a beekeeper, you need to keep them and take care of them. So and also, you know, with the viruses, the bees will just tend to just die, and you're not going to find many that are going to survive without any treatment. Bees are managed and they need to be cared for. So we are working on a trial on some VSH Varroa resistant hygiene bees, and there's many breeders that are working on this, trying to breed a better bee. These do take a lot of time and effort. The mating needs to be controlled really carefully. Some of them use instrumental insemination to control the drone stock. Because if you think about all the daughters from a hygienic queen, they have to go mate, and if they're mating with non-hygienic drones, you're diluting that trait right off the bat. So the mating yards for these need to be really isolated, which is really hard to find in our environment. And small scale beekeepers, you can try to raise local stock, resistant stock, but it's really difficult to do on a small scale. And also some of these bees are very resistant to Varroa, but if they're not production quality, if they don't make a honey crop, if they don't survive the winter, if they swarm really easily, if they're super aggressive, they're not really viable strains if they're going to be used for commercial beekeeping. So that's the trick, is to come up with a bee that's Varroa resistant and is good in other ways. And even with the Varroa resistant stock, treatments will still probably be necessary. They just won't be, you won't need to treat as often. That's the hope. So next, I'm going to pass it on to Nelson. He'll talk about some treatment options for Varroa.
Ready. Thank you, Ben. So hello, as Ben said, I'm Nelson Williams. I'm the Midwest Technical Transfer Field Specialist based at the University of Minnesota in St. Paul. And I'm going to be talking about the control options for Varroa destructor. So we certainly hope that you are using a scientifically verified and effective Varroa monitoring program. If your mite loads warrant treatment, please treat. So as we've said previously, treating is just a part of good animal husbandry. Let's say your dog has heartworms, which is a parasite. If you did not treat the dog, they could die. It would be the same thing for honeybees at the superorganism level. Without an effective control for treatment, colonies infected with high mite loads will most likely perish. Additionally, untreated colonies with high mite loads can become mite bombs, horizontally transmitting mites into neighboring colonies within their flight radius. So your problem becomes your neighbor's problems. So make sure that you're doing the neighborly thing and managing your mites. And then, as always, please follow the guidelines outlined in the Honeybee Health Coalition tools for Varroa management. There are a lot of unverified methods out there that claim to be effective treatments and controls for Varroa, but all of these methods, so the powdered sugar, small cell, screen bottom boards, mineral oils, essential oils, thermal regulation, they may have isolated anecdotal success, but they have failed to be backed up by consistent scientific research demonstrating positive Varroa control results. So there are a lot of mechanical Varroa control options, or there are mechanical Varroa control or caging queens, which will disrupt the Varroa's ability to reproduce and also offer a good window for treatment. So that's it. Yes.
Slides are back up. Thank you, Anne-Marie.
So that's the brood break, or is a good mechanical control to use in addition to the use of chemical controls to keep those mite loads low. And then additionally, as Dan taught us, an infiltrated drone brood cell can produce 3 to 4 mature female mites. So it is a good idea to remove drone brood frames and scrape that drone brood burr comb found between the boxes. Just scrape that off and get it out of there. So in addition to the mechanical controls, you may ask yourself which chemical Varroa control option is the best for me in my situation. There's synthetic controls, which are stable and safe to use with gloves, but they may struggle to knock down the high mite loads, while organic controls, such as acids and essential oils, will require additional safety measures due to the harsh and volatile chemicals used in these treatments, but you do have options. So when you are using any of the controls, always be sure to use the suggested personal protective equipment. So gloves, respirator, goggles, skin protection, according to the label instructions. Additionally, federal law requires any person who applies or supervises the use of restricted use pesticides to be certified in accordance with EPA regulations and state, territorial, and tribal laws. So none of the controls that we are going to discuss today require a pesticide applicator permit, but if you are using restricted use pesticides, be sure to visit epa.gov for further information and guidance. So what are the general rules for Varroa control options? First, I would start with not using coumaphos or fluvalinate because mites have developed resistance to these controls and they leave persistent residues in the comb. So you really don't want to infect your crop with coumaphos or fluvalinate. In an effort to combat resistance, make sure you are rotating your treatments. And once again, always use the control according to the label. And we're going to keep harping on this, but monitor your mite loads before treatment to deem if a control is necessary. And then once again, after treatment to verify the efficacy of the control that you use. So when you're selecting your chemical control, there are a lot of different variables in the equation. Most important is your Varroa load, so which can be determined by the monitoring. We prefer the alcohol wash. You also want to know which phase is your colony in. Is it dormant in the winter? Is it the population increase in the spring, the population peak in the summer, or the population decrease in the fall? Some of the control options available are temperature sensitive, so you want to make sure that you're checking the weather. How strong is your colony? Some control options can be rough on brood and queens and are best suited for the stronger colonies. Do you have honey supers on? If so, your control options are narrowed down a bit. And then what was the last control performed? Remember, it's best practice to rotate those treatments to combat resistance. So the most complete Varroa control guide is available at the Honeybee Health Coalition online. It's downloadable online. And additionally, they go into great detail on each control product in depth, which is really helpful. But why don't we click on the link here, and we can, it's just a really useful tool. We'll show you how it works here. After a few prompts to get started, we'll ask you a series of questions. And now we can begin. So first question, do your Varroa mite levels exceed the treatment threshold? Let's say I monitored and yes, they do. Are you open to synthetic and organic treatments or just organic? At this time, I am open to whatever will work for me. Is brood present? Yes, currently it is. It is, do I have honey supers on? So middle of July, yes, I'm gonna pull next week, but they are currently on right now. And is your colony's population increasing at peak, decreasing, or dormant? Right now, they should be at peak population. So I have several options. I've got the Mite Quick Strips, Formic Pro, and HopGuard 2. If you click on each treatment option, it'll go into further detail, talking about the active ingredient, application, just all the information that you need to know, in addition to the information that's on the label for getting that control onto your bees. We can go back to the presentation. Alrighty, so now I'm going to get into the different synthetic and organic control options available over the counter. So we'll start with Amitraz. It's found in Apivar, and it is a slow release synthetic contact control. Contact meaning the bees must come into contact with the strip to distribute it throughout the colony. It's safe and easy to use. For application, you would simply insert the plastic strip into the brood nest and anchor it to the top bar using the cut triangle that you can kind of see in that photo, or you can stick a nail through the hole and just kind of rest them on the top bars as it hangs down into the brood nest between frames. Amitraz is not temperature sensitive and can be used at any time of the year when supers are not on. So make sure that you are checking your calendar and giving yourself, a 7 to 8 week window before you super, if you plan on using Amitraz in the spring. The one drawback I would say with Amitraz is that it cannot be used with the supers, and it's not especially effective against high mite loads. The next option would be Thymol. Thymol fumigant, found in Apilife Var, which is a wafer, and Apigard, which is a gel. It's a slow release control that is safe to use with gloves and pretty easy to apply. If you're using the Apilife Var, you would simply put the wafers on the top bars at the corners of the brood nest for approximately 12 days, but make sure that you're reviewing the label here. And for Apigard, you would simply apply the individual dosage tray, which is about 50 grams per double deep. So once at day 0, once at day 14, or after 2 weeks, and then you would remove or spread the remaining gel over the top bars at the end, so 28 days or 4 weeks. So it's a 4-week treatment. Thymol-based controls are temperature sensitive and should not be used when supers are on. They're not especially effective against high mite loads, and you may see bearding or dead bees or dead larvae in the front of your colonies, but this is expected, just letting you know that the treatment is working. A few dead bees is letting you know the treatment is working. The next option we have is formic acid. Mite Quick Strips uses formic acid. It's a saccharide gel strip. It's a flash treatment fumigant that requires acid-resistant gloves, so nitrile gloves. You're going to want to wear skin protection, and it's not a bad idea to use a respirator. The fumes off of the oxalic or the formic acid can be pretty harsh. So a full dose is 2 strips placed on the top bars of the brood nest for 7 days, or you can do a half dose, which would be 1 strip for 14 days, followed by a second strip for an additional 7 days. So a half dose is a 21-day treatment. And when you do apply the formic acid, you may want to ventilate the colony, so offsetting the boxes or adding a shim or drilling a hole into your deeps to get that ventilation in there, because the fumes are a little bit rough. It's safe to apply during the population increase, the population peak, and decrease. Safe with supers on, and can be effective against high mite loads. It also works under the capping, so that's it's a benefit to using the formic acid. The few drawbacks, there's a chance for bee brood mortality and queen loss, and it is temperature sensitive, so make sure you're checking the weather. Once again, you may see bearding, dead bees, dead larvae, but this is expected when using formic acid. Next, we have hop beta acids. So that's found in HopGuard 2, and I believe there's a new HopGuard 3 out. It's a natural contact control and folded cardboard strips. They recommend that you use gloves and eye protection when applying. So one strip of the HopGuard for 5 frames of bees in each brood box. It's a 4-week treatment, and you don't want to use it more than 3 times a year. It's safe to apply during the population peak, the population decrease, and dormant periods, and it also can be used with supers on. Few drawbacks: the strips are a little bit messy, and this one, we suggest that you do a check back after all of the control treatment options, but especially with HopGuard or hop beta acids, you want to make sure that you do a check back to make sure that the application is working. One note with HopGuard, I generally like to use them on smaller colonies or colonies where I don't want to disturb the queen. It's just it's less disruptive to the colony behavior. And the last control option that we're going to discuss is oxalic acid. So there's 2 different methods for application. There's the contact control, which is the dribble, or the fumigant sublimation method. With oxalic acid, we recommend that you use gloves, respirator, skin and eye protection. So you can see Anne-Marie on the right there, all geared up with her gloves. That's for the sublimation, and then the photo on the left is the dribble. So for the dribble application, you mix 35 grams of oxalic acid into 1 liter of 1-to-1 sugar syrup, and syringe about 5 milliliters directly onto the bees in that bee space between the frames. And for sublimation application, you would use 2 grams per hive, following the label and the vaporizer instructions. This is best used during dormant periods when there's little or no brood, and due to it's a bit can be hazardous, is hazardous to human health. So make sure that you're using it according to the label and that you're using personal protective equipment. So it's a really good treatment for a broodless dormancy winter cleanup. And also, there's beekeepers that apply it in rounds 7 days apart, probably 3 rounds, 7 days apart in the fall to get rid of those mites on bees. So this, we get a lot of questions of, well, what should I use and when should I use it? So we put this slide together just as kind of a general directional schedule. So the population increase in the spring, we definitely want to start with the mechanical controls, splitting, requeening with hygienic queens, drone brood removal, and then you have the options of amitraz, thymol, formic, and hops. During the summer or the peak population, with supers on, your options are narrowed a bit to formic and hops. During that population decrease in the fall, amitraz is once again available, thymol is once again available, and then you have formic and the HopGuard. And then in the winter, the oxalic dribble and vaporization is a good option. So this last slide is the control options cost for Varroa. So cost is a factor when selecting the control, but you really have to consider the cost of not treating, and that would be unsustainable colony losses year after year, and then more money out of your pocket to replace those losses. So, I'm not going to go into the details on which product is most expensive or least expensive, but just wanted to give you an idea of what it costs to treat one colony, estimated at 15 frames of bees. So having walked you kind of through the Varroa history, biology, population growth, monitoring, and treatment options, that we hope that you start or continue to monitor and control for Varroa. And I will pass the mic to Net to wrap up this webinar with an exit poll.
All righty, so this last poll question has 2 questions this last poll has 2 questions. So we'll let this run a little bit longer. We'll give it another 15 seconds here. Okay, we have about 80% responding. I'm going to go ahead and share the results and talk through them for the sake of the recording. In the top question, how often do you plan to monitor for Varroa? This has changed a little bit. The once per year went down, and so that's at 2%, twice per year at 32%, at least 4 times per year at 40%, every month at 24%. I do not plan to monitor at 2%. And then when we look at the methods, they were a little bit more spread out. Alcohol wash didn't have as many. It looks like the webinar might have convinced some folks. It went up to 73%. Visual inspection of adults, 23%. Visual inspection of drone brood, 29%. Sticky border mite drop, 29%. Powder sugar shake, 27%. Ether roll, none. All righty, so I'm going to stop sharing. There we go. All right, well, I want to thank all 3 of our field specialists for joining us today. If they could hear your round of applause, it would be loud, I know, because I've gotten a bunch of questions in, even though I requested that you send them elsewhere to the Ask Questions Here box. I've been trying to forward them and keep up, but I know we might not get to all the questions because so many have come in, and that just reflects all the important information that our presenters are sharing. So a big round of applause to our 3 presenters today. As I like to mention, we are a nonprofit. If you are interested in helping us out and supporting us, we do have a donation link up on our screen here. We are going to be doing another webinar in 2 weeks. This one was recorded and will be on our site within a day or so. And you can always check those out at beinformed.org/education, where we keep all the recordings. We do have a new YouTube channel where those are stored. So if you subscribe to our older YouTube channel, the newer videos have been moved over. And so you can subscribe to our new one, and you'll be able to find that through going to any one of our videos on our education page on our website. So at this time, I'd like to turn it over to Natalie Steinauer. She's our science coordinator. She has been collecting a ton of questions, probably sorting through and combining some. I've asked her to pick and choose ones for the good of the group. And so I will turn it over to her at this time.
Yeah, so we received a lot of questions. Thank you so much for your interest, and I've grouped them into broad categories. So we've received a lot of questions on the life cycles of Varroa, testing and treatment. So I'm going to start with a couple of questions on the life cycle. One of the questions was, that the hygienic behavior that bees uncap the cells, they were wondering if how did that really kill the founder mite, or have the mite just find another cells to move into, and so what was the benefit of that hygienic behavior?
Yeah, so that uncapping, it's correct. It's not going to actively kill the mite. It's limiting the growth rate. It's disrupting the reproduction. So while that founder's mite will still survive, she's not coming out with 1, 2, 3, 4, daughters alongside. So it's a slows the growth, behavior, not a destroying the mites that are there.
Thank you. Other question on the life cycle of the mites is it so you showed a picture of the PMS, and one person asked, how do you differentiate that PMS melty look from melty look of larvae with infected with European fall brood? And also, is it possible to cure a colony once you see this in your colony?
Sure. So as far as differentiation between European, because you're right, there are, a number of stressors on a colony that will lead to kind of some deflated, melted larvae like we saw there. One way I help differentiate is kind of context clues. A mite wash, a number, if I'm getting a lot of mites out, if I do a wash and I get quite a few out of a sample. If I'm also seeing bees with deformed wings, that's pointing me towards mite caused. I've actually, if it's that severe of an infestation, maybe actually seeing mites on bees. We talked about it's not a great method for low-level infestation detection, but when we're seeing a colony that's under that much stress, we probably, with careful examination, would see mites on bees. And then certainly also looking at frass in cells. If you kind of rotate that frame so the top of the cells are pointing away from you where you can see the ceiling, you would expect to see frass in a number of those cells. So those are kind of the context clues that are going to point me towards mite damage. And as far as if you didn't see any of those, you were still not sure on differentiating. One thing I've kind of become accustomed to using when I'm not clear on a diagnosis, just from visual inspection, there are now available field test kits from the, I think they're available through all the bee supply companies. They're about $15. They come for either AFB or EFB. And in using those, you swab a little piece of a suspect larvae, you put it in a solution, shake it around for about 20 seconds, and then put a drop on a test strip, and it will tell you whether that is EFB or not. So sometimes those are useful. We actually go through those. A previous webinar, we did brood diseases where we talked about European fall brood and using those. So I would encourage you to look at that webinar for more details on that.
Thank you, Dan. All right, moving on to testing. Mostly the question that we receive from multiple people is about the necessity to monitor while you have decided that you are going to do treatment. So some people say, I know I have mites. Why do I need to testing? What do I need to do testing to monitor if I have already decided that I'm going to use a treatment?
Yeah, so some of the commercial beekeepers, they have a certain schedule that they, treatment schedule they're planning ahead. But the reason for monitoring is first of all, if your mites are at 0 across the board in all your samples, you could save some money by skipping a treatment, and that's not too common. But there are times when, if your fall treatment was really good and you tested early in the spring, you might get 0 mites in all your samples, in which case your treatment would be it'd be better to wait on that and save the money. So just knowing what's in there it's important, and then the follow-up is even more important, that testing, sampling for mites after your treatment. So each of these treatments, they have their pros and their cons, and there's definitely been times when we look at a colony after it's been treated and there's still mites there. For whatever reason, it didn't work right. Maybe the temperature was wrong. Maybe the colony, the dynamics within that colony was off, or something was, if you're fogging with oxalic acid, if there's a lot of burr comb and a lot of obstacles in the hive. Maybe it didn't circulate totally well, or maybe you place the treatment on, on top under the lid when it would have been more effective between the brood boxes. So there's all these variables that can affect how effective the treatment was. So just that follow-up testing is really important, because if you, for whatever reason, your treatment didn't work, maybe it was expired and it just was denatured. So that follow-up treatment, if anything else, it gives you peace of mind that, okay, that treatment worked, my mites are low, I can sleep at night. So
And then a follow-up on that, one person asked, how long after the control should you actually measure for efficacy?
That's a great question, and I've thought about that, and I've heard a few different answers to that question. But I think it's good to wait at least a week after that last treatment. Maybe even better to wait a little longer, 10 days or so. We want to give that, because that mite treatment, whatever it is, chances are it's killed all the phoretic mites, the mites that are not in the brood. So every single day, there's more bees hatching, there's mites emerging. Some of the treatments, they linger a little bit. So you might get some mite drop 4, 5, 6 days after that treatment, especially the longer acting ones. The Apivar strips are supposed to last for 6 weeks. But as those mites are hatching every day, some are dropping, some maybe are not. So I think you want to give a chance for that brood to hatch out and see how many mites are still remaining. That's why I think it's really crucial to retest, and then also be prepared to put another round of treatment on, especially these flash treatments. They'll do a great job of killing all the mites on bees. But even if you see the treatment in a week later, there's still a remnant of it left. That might not be killing mites that long after. So I think to be prepared with another round of treatment is really important. And a lot of times commercial beekeepers they'll reply 2 or 3 rounds of each treatment, just to, as that brood is hatching, to get those mites as they emerge.
Another question that we received in terms of testing is it necessary to sample every hive, or how do you sample, how many samples do you take from each apiaries? And then kind of like a follow-up on that is, if you have one colony with a very high mite load that you want to treat, should you treat that colony punctually, or should you just treat the whole yard?
Those are great questions too. So we did a trial way back at the beginning of BIP about looking at how many colonies per apiary we need to look at, and the magic number that we came up with was 8. So if up to 80 or 100 colonies in the apiary, if you look at 8, that'll give you a good representation of what's in that yard. So that's what we tend to do. If there's over 100, we'll tend to look at about 10% of the colonies in that apiary, and it'll give you a good cross section of what's in there. And if you just have a few colonies, I mean, if it's not too much trouble, I would test every one if you have the time. And the question about if you find one mite bomb, whether to treat that one or the rest, again, I've heard different opinions about this. There's the argument you've heard of prophylactic treatment, which is preventative treatment, and then there's metaphylactic treatment, which is you notice the problem in 1 or 2, and then you blanket treat, because chances are there's developing issues in the other colonies. So I tend to go with that second method of even if you have one that's really high, I would say it's probably not the only one in that yard. I would probably treat them all, so that you're not missing any. And another thing to note is that sometimes you'll get colonies that there's waves of mites. If there was a brood event or a queen event, let's say the queen stopped laying or was requeened, you have all these mites that are just hanging around waiting for that brood of the right age. And then when that queen starts to lay again, they all dive in at the same time or within a couple of days. And then 10 or 12 days later, you get this big surge of mites. So if you tested, soon after those mites crawled in, you might get a really low mite number. And that's kind of disguising the true number of mites. And if you test right at that 10th day after that queen event or 12th day, you might get a wave of mites that are all hatching at the same time with that first round of brood. So that's why we take multiple samples out of a yard, because it gives you an average. If you just take one or two, you could be misled about the levels in that yard. So hopefully that answered your question partially, at least.
Yeah, thank you, Ben. Another question on testing. There were a few comments on the percentage of alcohol that we recommended in this presentation. Some references to Randy Oliver research, showing that 91% alcohol has a higher recovery rate of mites. So can you give us some more details on that?
Yeah, I totally believe that. I've noticed that also. It seems that the higher percentage of alcohol is, the more mites you get out of that first wash. And that's exactly why we rinse those bees many times, not just once, but at 35% alcohol, the mites are still dropping off, but you want to do it a few times, because a lot of times you'll see on the first wash you might get 4 or 5. Second wash, 1 or 2 more. Third wash, maybe 1 more. And I pretty much do it until I get zeros out of that sample. So it's true, 91% might get more mites off that first rinse, but that's why it's important just to rinse them a few times if you're not using that full strength.
Yeah, absolutely. And I would like to add that one of the reasons that we have decided as a team to go with a lower concentration was also for safety reason for our tech team in the field. But that's again is why we're, our tech team are doing multiple rinses rather than a single one with a higher alcohol content. All right, so moving on to actually treatment, there was a few questions on alternatives to treatment. For example, the Varroa sensitive hygienic and drone brood removal. So a couple of persons have asked a question about the scraping the drone brood, and if there are any comments on the efficacy of doing that, as well as brood break. So basically, can you talk a little about those alternatives, brood break and drone brood removal?
Yeah, sure. So the brood break, either caging the queen or making splits, essentially you want to create an environment where the queen is not laying for probably about 2 weeks or so, because that would allow the capped brood to emerge, all of the mites to emerge with that capped brood, and then give you a good window to attack those mites on the bees. So, and just removing the drone comb, I think it's just good beekeeping practice to scrape that burr comb out of there. And as we saw with the extended capping time for drone brood, those extra 2 mites that may emerge with the drone brood, you're really cutting into their numbers. So I don't know if Dan or Ben want to add anything there, but.
I would just add that I think they can be helpful, but they're supplemental. I'm not, I haven't seen a lot of evidence, particularly from new beekeepers, that they're finding any measure of success exclusively using those methods. I look at it as it doesn't hurt, it's worth doing, but they're not going to be sufficient on their own.
I would second that.
So we had a couple of people really asking specifically regarding to hot weather treatment. So you have covered temperature limitation in this talk, but some people really just wonder when you are at a higher temperature and you do have to treat, what options are remaining. I think we do, we've covered that, but maybe can you just pinpoint them to a simple answer here?
Yeah. So I'm from St. Louis, and it gets really steamy, especially now at this time of the year. But I know that I'm going to need to treat. So that's why I use the formic acid, and there was 2 different doses. So you could do a 2 strips for 7 days, or you can do 1 strip for 14 days and then follow up with a second strip for 7 days. I feel like the stress from that half dose, so the 21-day treatment, 1 strip 14 days and another strip for 7 days, the bees seem to tolerate that just a little bit better. Seems like it's less stress on the brood. I'm seeing less brood mortality. The queens seem to bounce back a little bit quicker, just because you're not that dose that you're using is not as hot. But yeah, it's another question that I'd probably see if Ben or Dan had anything to add.
Yeah, I would say it just brings home the point that it's if you can keep your mite levels lower in the early part of the year, you might not have to worry about treating in midsummer. So I would say think ahead and try to, before you put your supers on, you have much more options. So I would put a treatment on in the spring if you have mites there, and then you can probably, if your mites are low throughout the summer, wait until the honey supers are removed later in the year, and then treat immediately and aggressively after the honey is off. And then again, it should be cooler and you should have more options. So I think planning ahead can go a long way.
Yeah, I would certainly agree with that, and it may it may push you to decide to pull honey earlier. And if you're in a place with fall honey, it may be necessary to just forgo that and let the bees, gather that for winter food. If it's, if the alternative is leaving it on while they continue to build mite numbers and kind of enter a real bad state of health. But yeah, I think certainly more proactivity in the spring to get as close to zero as you can before you limit your options with supers and temperature.
Thank you. A follow-up on that was a question about the restriction for honey supers. If it also, concerns any honey that you, for example, in top bar colonies where they don't really have a super, but they have frames that are honey that is mixed with the brood. Do you consider that, honey that might be extracted and therefore subjected to the same limitations? Or how do you deal with honey that you do not plan to extract, or those type of conditions?
Yeah, if it's a top bar hive and you're planning on extracting that honey, I would apply the same rules for Langstroth hives. If you're planning on extracting that honey, then I would follow the labels on what is acceptable to use with supers on or honey to be extracted, and which controls are not acceptable to use with supers on or honey to be extracted.
Thank you. All right, and I'm going to finish with just very practical questions about the monitoring. Some people ask about why you use a cup and not like 2 times a quarter cup and not a half cup, and also the size of the mesh size of the screens that you're using for your test.
Okay, so that so the quarter cup measuring scoop, that's Dan Orell's preferred method. I think he thought he could get a more accurate bee count. But if you want to, if you can use like a spam can or some kind of other container and measure half a cup, I think that works just as well. But you just have to make sure that as you're collecting the bees, you kind of tap it down so that they settle, so you don't get a low bee count. In our Sentinel program, we have beekeepers taking their own samples, and a lot of them are come back real light. They should have 300 bees, and some of them have 150 or so. And if you use the 2 quarter cup style, Dan thinks you get more accurate count. But I think the half cup measuring cup works just fine. And sorry, what was the other part of that question?
The size of the mesh.
Oh yeah, so I believe the mesh for, I want to say it's 3/8 of an inch or it's smaller than that. It's but it doesn't really matter as long as the bees aren't falling through. and it shouldn't be small enough that they're snagging mites. So a standard sieve is usually fine. And with the mite capturing screen, that needs to be much finer. So like something like a window screen mesh or smaller, preferably smaller mesh that's going to filter out the mites. And you can find these in a lab supply store or even the grocery store. If you look, you can find sieves that have the right size mesh.
Okay, I think we have covered most of the questions that came in. If I forgot your question, I apologize. We received a lot of questions during this presentation. And so, yeah, don't hesitate to email us if you have questions that have been left unanswered. I just try to keep it to general questions rather than particular instances of recommendations. But,
Great, thank you, Natalie. Yeah, we did get quite a few in, and what we may do is sift through them. We can save this chat box and see if it's if there's enough themes that we may do a blog down the line. So if you haven't subscribed to our blogs, that's something else. You can go onto our website. There are a lot of subjects on there that you can search, and you can get on that list to receive those if you're interested. We also send out announcements about the webinars to that list, as well as any past registrants to webinars. You'll get a notice about a new webinar. For those of you who registered for this particular one, we will send the recording out once we have it finished. And at this time, I'd like to thank everyone for joining us, presenters and audience, and we really appreciate you supporting us through this. Everyone stay healthy, and we will see you on another webinar in the future. Thank you so much.